All right, good morning, everyone. We're going to go ahead and get started. We have a lot to cover today. Thank you so much for being here for our webinar on surgical neck. We work really hard at the NEC Society to keep patients and families centered in everything that we do. So we're really honored to dedicate today's webinar to Sweet Reed. Reed was born at 25 weeks gestation, and for the first several weeks of his life, he was doing really well. His family was really excited to look forward to the day of bringing him home. But unfortunately, Reed was diagnosed with neck, and despite the excellent care from his medical team and his family's love that they surrounded him with, and despite the surgery that he had and the treatment, Sweet Reed did pass away from neck. And so we're really honored to dedicate today's webinar to him. And we're thankful to his family for being involved in the NEC Society and for giving back in honor of their son in such an impactful and powerful way. So thank you for keeping Reed in your hearts today and all the families that have been affected by this really devastating disease. And speaking of infant loss, October is Infant Loss Awareness Month. And tomorrow, October 15th, is Infant Loss Awareness Day. So at 7 p.m. local time, we're going to be participating in the Wave of Light, where we will be honoring babies and remembering the little ones that we carry in our hearts who have passed away. So if you are able to join us for that, we will be lighting candles tomorrow evening. And if you are just getting to know the NEC Society, welcome. Thank you so much for being here. The NEC Society is a 501c3 nonprofit organization that's dedicated to building a world without neck. We bring together families who've been personally affected, clinicians, researchers, and other people who care to advance neck research, education, and advocacy. And the photos here are from our meeting in 2019 at the NEC Symposium that was hosted in partnership with the University of Michigan in Ann Arbor. And we're really excited to be bringing everyone back together in person next year after we have been virtual for way too long because of COVID. So looking forward to seeing everyone again in the not so distant future. A lot of the work that we've done over the last couple of years has focused on identifying the most urgent and hopefully impactful research questions and topics that matter most to people like you and people like us here on this webinar today. So here are the NEC research priorities that we are going to be dedicating much of our resources and time to advancing in the coming years. So here are your wonderful presenters that we have for you today. We're really excited for this lineup of speakers. These are all pediatric surgeons that were so dedicated to helping families improve outcomes and provide the best care for babies that have been affected by this really devastating disease. I should start by introducing myself if I haven't had the pleasure of meeting you. Again, thank you for being here. I'm Jennifer Canvasser, and I'm the founder and executive director of the NEC Society. I lost my son Micah to complications of NEC just before his first birthday, and I feel really honored to be able to do this work here at the NEC Society and to be able to work with all of these amazing people who you're going to hear from today. So first you'll hear from Samir Gadapali, who is actually Micah's pediatric surgeon. He's at the University of Michigan. Kathy Hunter is at the University of Oklahoma. Colin Martin is at the Children's Hospital of Alabama. Troy Markle is at the Indiana University Health Center, and Gail Besner is at Nationwide. So really excited for this lineup of speakers. Today's learning objectives include a few of the key points here. We hope that you will understand the types of surgical treatment options that are available for infants that are diagnosed with NEC, understand what surgeons use, the kind of criteria that they use when deciding which treatment options are best for the babies that have been diagnosed with this disease, and then just describe some of the recent evidence-based projects that are taking place related to surgical neck patients and their families. Just a friendly reminder that we are never providing medical advice and that all of our faculty views are independent from the NEC Society as an organization. So we're going to go ahead and get started with Samir. I'm going to hand it over to you, Samir. I'm going to stop sharing my slides and you are up.
You should be seeing my screen now.
Yep.
Perfect. All right. So thank you for having me here today. I'm honored to be one of the guests in this group. My story follows the story of the NEC Society very closely. And the reason for that is because When I was a pediatric surgery fellow in the last year of my training, I actually met the Canvas family and our two boys, Micah and Zachary, in really difficult times. They were both diagnosed with NEC, though Micah needed surgery, multiple in fact, as he required bowel resections and an access for dialysis. When I graduated my fellowship, I became an attending at Michigan and covered the ICU as a faculty member. Micah was on my service for several months, and when he passed away from complications of NEC, I was actually there for his life celebration. And I can't stress the impact that his hospital course, his life, and his family have had on my training, my career, and my life. Approximately a year later, I became part of the NEC Society and helped with the study of the family members impacted by NEC that were part of the society. We found that we kind of needed to do a better job educating families earlier about the risks of NEC, communicating with families about the importance of breastfeeding, and most importantly, empowering and partnering with parents in the care of their children. No longer was a paternalistic approach to care acceptable. We published our data in several forums and eventually published the manuscript. This was the first of several research projects that I learned to partner with families. It's kind of starting to do research with families rather than on families, which is an important distinction and goes along with much of my career. The next step in my journey led me to the first NEC symposium, which was held in UC Davis in California. As a junior faculty member, I was involved in the planning of the conference and had an opportunity to deliver a talk on the surgical approach to NEC. All of the main findings were summarized in the Seminars of Pediatric Surgery journal published in 2018, where Dr. Markle was one of the editors, and you'll hear from him in a bit. There was an article in that edition that focused on the development of the NEC registry. That first conference is where Dr. Good, myself, and others decided on changing the paradigm on how we collect data for NEC. Traditionally, most scientists use samples available to them in their own hospital systems to study the processes that underlay NEC or other diseases. We wanted to amplify the sample collection and collaboration among the scientists from across the country. I continued on my path for clinical research in NEC and learned about kidney injury and differences in NEC for surgical management, as well as preterm and term neonates, and had several publications related to that. My largest contribution to NEC has been to host the second NEC symposium held in Ann Arbor. I had the opportunity to work closely with families on applying for funding, developing a budget, webinars with scientists, funders, families, and invitations to the same scientists, funders, and families. This was an all-in meeting where parents and patients were also the hosts. There were the speakers, there were the panelists. All of the sessions were dedicated to children, patients who had the disease. They were the first to ask questions. They presented their posters. And this is the proudest I've ever been for any scientific conference. I learned so much from families during this time. And what I learned was, one, science has to be understood by everyone. If we really wanted to improve where we're going in our society, it's not just clinicians and scientists and researchers that need to understand, but patients and families are so important to help move the field forward. Second, the language that we use is super important. Certain things just automatically make you think a certain pathway, and just changing the language of how we talk helps you reconnect and rethink about ways to do it a lot better. And finally, advocacy for NEC. Family-centered care was not the same as patient-centered care. Parents are care partners, and NEC is a family disease. Considering patients or neonates, it's a family condition and experience. And we always think about the quality of patient life, but really it's the quality of the parent's life is just as important, and the family life, and how much this disease affects not just the child, but also the entire family unit is very important to think about. And we also learned a lot about empowerment of families in research. I think the mindset that we use in terms of conducting research has always been, we collect these samples from families, and a lot of times they are the ones collecting the sample. There was one mom that explained how she took a sample of stool at every appointment for a study, and the amount of work required for these families, and they're happy to do it because they want to like make things better. However, she didn't even know what happened with the study. What were the findings? What side were they even randomized to? And I think that's just not acceptable. And then we also found that a lot of funding agencies don't really have a family representative. So how do we decide what's really important to patients and families? We learned that money dedicated for NEC research was so infrequent and small compared to the amounts given to many other neonatal conditions, despite the devastation it leaves behind and the catastrophic effect it has on all these families. I'm going to skip the highlight video for the symposium for time purposes. In conclusion, what I learned was parents and families are valuable partners in research. They help me with developing research questions, funding, recruitment, and even disseminating findings. Advocating for me and my research, as well as others in the institution, nationally and internationally. It's a way to solve really complex problems, and it's not something that is utilized enough. And most important team member is not the researcher, but the researchee, the people we're actually doing the research on. Thank you for giving me the chance to present.
I love that. Thank you so much, Samir. Really appreciate you highlighting everything that you've done with us. Kathy, you ready?
I am. Well, again, thank you so much for this opportunity. I hope you're seeing my slides okay.
Yes.
Okay, fantastic. So it's an honor to be part of this panel. I have nothing to disclose. As Samir was saying, the whole point of what we do is for the patients and the families. For me, I remember I was a medical student in New York at the time, and I was on my surgery rotation, and I happened to have a chief resident who was planning on going into pediatric surgery himself. And so we got to sometimes go and visit with the pediatric patients. And I remember when the first time in the NICU, I met a little baby who had necrotizing enterocolitis that undergone surgery. And I was really struck, first of all, by being a person who'd never been in a neonatal intensive care unit, how tiny that baby was, and moreover, how we were able to try and help that baby. And it seemed so fragile, and it also seemed so amazing. And it was really just something that was incredibly inspiring to me. And every single day, I think one of the things that makes this job so worthwhile is that we really do get to learn from our patients, and we try and have an impact for these patients and for their families. And just when you think that you maybe have a particular way of doing things, you always need to challenge yourself and need to ask the questions and be able to be a constant learner. I decided that I was going to be a pediatric surgeon, and I spent several years in California at Children's Hospital Los Angeles. I worked with Dr. Henry Ford, and I was in his lab actually studying a bacteria that I think you've probably heard of at this point, Bacillus sakazaki. It was one of the pathogens that was partly responsible for that shutdown of the formula plant in Michigan recently. And that was something that that lab and myself were looking at in 2007. I think the thing about NEC is that it really is so cruel that it affects our smallest babies. It really can be devastating. And we just want to have a multi-pronged approach to try and eradicate this. The clinical work and studies are imperative and important to be directly at the bedside. Myself and some of my other colleagues on here today, you'll hear also how we have tried to use basic science approaches to try and define this disease. We know it's complex. We know it's not exactly the same for every one of our patients. We know that there might be prenatal, postnatal, other environmental factors which might be predisposing, but we really don't have all the answers despite years of research. We really want to be able to find ways to predict NEC before it happens. And then importantly, we also want to identify ways to try and stop NEC once it's already started. And then trying to determine strategies to improve our outcomes and also improve our survival for our patients. As a surgeon, I often think that I'm able to make a difference for a person at an individual level. And the exciting thing about research is that I really feel like we're trying to make a difference, not just for the individual, but also for the entire population. I'm going to give you a few slides about the basic science research direction that our lab focuses on. In the intestine, there's an intestinal barrier. This is a really important barrier between that external luminal world and also that internal world of the baby. We know that babies that have been born early have a premature intestinal barrier, and sometimes they're a little bit more leaky. We know that barrier is just imperative to trying to maintain a healthy relationship between that microbiota and the actual immune system of the body. In fact, the gut is the largest immune structure within our bodies. Permeability of the intestinal barrier is regulated by these little connectors that are called tight junctions. And they're small complex proteins that actually regulate this passage between those cells and barrier. And it's something that our lab has been very interested in trying to see if there's intrinsic differences or susceptibilities in some of our babies versus others. Again, you don't need to understand all of the pathway stuff, but what we did is we helped identify certain kind of chemical pathways within the cells that are able to affect changes in these structures that bind the cells together. The one that we've been looking at most recently is the rho-associated protein kinase pathway. It's a particular type of serine threonine kinase protein, and it's involved in a lot of things within the cell, including those tight junction regulating factors, contraction of cells, and also regulating how cells live and die. There have been studies in humans before looking at how the inhibition of the system can help with the leakiness in other cell types. And so what we've been doing is looking at whether or not it's actually activated in NEC. So again, with the generosity and kindness of families who do help, they inspire us, and then we also agree to allow some specimens to be used as study samples. We have identified that there is an activation of this protein in some of our human samples. We've been able to look at this with protein levels, as well as looking at it within the immunofluorescent studies that you can see below there, where red is lighting up, and it's that rock chemical that I was talking about. We also work with cells in lab tissues. These are some in vitro studies showing similar findings. And then the thing with necrotizing enterocolitis work is that when you're trying to test whether there's something that might be therapeutic, you need to have systems that you can do that in. And a lot of us have worked in the past with systems including rat and mice models. You can see here that we used a ROCK inhibitor in an animal system. You can see that in the clean sample, the structure appears pretty normal, and if you compare that with that NEC sample, everything was kind of broken down. We saw improved health of the intestine with the addition of our inhibitor. And we also saw improved survival of the cell types within the samples when the ROCK inhibitor was present, as opposed to that bright red area, which is shown that experimental necrotizing enterocolitis. And this also corresponded with some of the changes in the tight junction protein called the gluten, which you can see was less bright in the NEC sample and much brighter when that ROCK inhibitor was present, more similar to those clean or healthy samples. A lot of us have been working to try and develop better models of disease, and we've actually been able to take some samples from discarded tissue samples from some of our patients and generate almost like little intestine balls in a Petri dish so that we can use in model systems to try and evaluate the effect of some of these chemicals on real human tissue without exposing any of our patients to real life risks of having experimental treatments in an already fraught situation. This is just an example of how we harvest and then grow these little intestine balls, and we can work with them in our laboratory settings. So one of the questions that we had to sort of think about today is, how is being a surgeon scientist affect my approach to things? Well, I think that whenever you have time to sit and think about it, I do feel like I have a pretty decent understanding of our current understanding of pathophysiology of disease. I work really, really hard to try and understand some of those associated risks and sequelae of NEC. It's not just an acute event. this disease has repercussions that are lifelong for families and the patients. In research, we really do want to look at the population as well as the individual, and try and have impact as broadly as we can, while still maintaining respect and the utmost regard for individual patients. And I think that there is a lot of exciting research going. I know that it was mentioned that our funding at times isn't as generous as some other diseases, but I really do remain hopeful. I've been pretty fortunate to have had funding for a variety of different sources, which has allowed me to drive some of my work forward. And you hear from the others today regarding their work. And I don't underestimate NEC. It can be devastating, it can be quick, and it is certainly unfair. And thank you for the opportunity.
Thank you so much, Kathy. Colin, you're up.
Can you guys see my slides and hear me okay?
Perfect. Yep.
Great. It's a pleasure to be here, and it's an honor to share the panel with friends and colleagues, and this event by the NEC Society is really amazing to be a part of. So I'll start with a case presentation, and this case presentation is probably unique to all of us on this panel. A 6-day-old baby who is extremely premature, weighing 900 grams, had clinical symptoms of NEC. But I want to highlight this is from a 16-year-old African-American mom with delayed and limited prenatal care. And so this is what the intestine looks like often when we operate for necrotizing enterocolitis. And because of the work that many of us have done on this panel and others, including families and advocacy, we have improved survival for babies with necrotizing enterocolitis. But despite all that work and understanding the latest techniques, there's still a pretty significant mortality and morbidity. So I'm a pediatric surgeon at Children's Hospital of Alabama. This is the children's hospital that is affiliated with the University of Alabama at Birmingham. And some of you have been to Alabama, but we have about 5 million people in our state. Our cities a little bit over 1 million. We have a pretty big children's hospital and pretty big based on square footage, and it's also pretty busy. And what makes it busy is that there are very few children's hospitals in the state of Alabama. So we're in Birmingham. There are a few pediatric surgeons who are close colleagues and friends in Huntsville, then also a few down in Mobile at the very bottom of the state. And so a state of 5 million, a lot of patients have to travel a great distance, and there are very limited resources throughout our state. So these are maps from looking at access to maternal care. So the darker the color, these are considered maternity care deserts. So these are places where there are very few OBGYNs or prenatal care within the county where patients are living. And the blue kind of highlights the Southeast and Alabama. A very similar trend for underinsured. And this is not to be a political map, but there's more women who are underinsured sort of in the Southeast and Alabama. And then looking at the prematurity score. So Alabama gets an F for prematurity score. So you think about access to maternal care, women who are underinsured, and prematurity scores. You think about the environment that these babies are conceived in, how that might affect their immune function. So going back to that case report, I gave a case report of a 16-year-old female who's African-American. This is an actual case. And so this mom was 16, was not in high school, had very little parental support, very limited access to resources, et cetera. So compare that scenario to someone who's 25 or 26. My wife and I were blessed with 4 healthy kids, and we had our first child when we were 27, and I was almost done with my training. So we had access to resources, we had family support, maybe too much family support. So compare those two scenarios. And so the women in the case scenario that I mentioned, all these things: partner violence, age, humiliation, danger, lack of employment, lack of housing, access to care, all those things makes her environment really, really stressful. And so our hypothesis of our lab is that psychological stress during pregnancy may disrupt the intestinal immune system. And this may be a cause of some cases of necrotizing enterocolitis. So there have been a lot of talk on this topic. This is a recent article from the New York Times about stress during pregnancy. And just to sort of highlight some of the excerpts from the talk, just kind of reading from the bottom panel. Women in America, an inescapable atmosphere of societal and systemic racism can create a kind of toxic physiologic stress resulting in conditions including hypertension and preeclampsia that lead directly to higher rates of infant and maternal death. And so just a few topics just to preference the rest of our talk, what are our lab studies. And so microbial colonization, this is colonization with good bacteria. Is very important for intestinal immunity and development. And also the immunoglobulin IgA. This is a immunoglobulin that helps to ward off harmful bacteria. When babies are first born, the IgA comes largely from breast milk. And then after a couple of weeks, the babies have the capability to develop or produce their own IgA. So in the context of stress and what's going on in the maternal environment, a seminal paper coming from Washington University in St. Louis showed that if you look at the stool from babies on day of life 0, so they're born and you get a stool sample, and if you follow the babies that go on to develop necrotizing enterocolitis in a week or two, going back to those stool samples, the microbiome and bacteria is different at birth from babies who develop NEC. So that suggests that perhaps there's an upstream insult, something's going on in gestation or the environment that might be playing a role. So our lab, we've created a few animal models, and these animal models were subjected to very tight regulation to be sure that our animal models were humane and done in a correct way. And so we have an animal model of pregnancy during stress. And so we have a mouse model where mice are stressed during their gestation. So the gestational time period from when mice conceive, when they have pups, is about 21 days. And so sort of during the second 2 trimesters of pregnancy, we subject the animals to different stressors. And then we look at the pups after they're born. We also developed a zebrafish model using the same thing. And zebrafish also have a lot of advantages in using them as an animal model. So just really briefly, some of the work that we've been able to accomplish in this space has been really informative for our lab and has allowed us to collaborate with a lot of other labs in this space. But I will highlight one paper that we recently published to look at some of the data that we're looking at in this space. So in this paper, again, our animal model was that when we took mice who were pregnant, we stressed them during their pregnancy. And during stress, mice that did not receive stress, their IgA levels went up. And the animals that were stressed, there was no change in the IgA levels. So IgA should go up during stress, but during pregnancy, but during stress, there was no change or maybe a trend towards a decrease in IgA. This is somewhat of a busy slide. I just want to draw your attention is that not only is IgA different in stress, but this is looking at now we stress the moms, and now we're looking at the pups or the babies that came from the stressed moms. And we see that the IgA binds to bacteria in a different manner between stress and control. We look at the microbiome, and there are microbiome differences in pups that came from stress dams compared to control. And we wanted to further dive in to see what those microbiome differences were. And we found 2 bacterial classes that were different in pups that came from stress moms compared to control. So Lactobacillus is one bacteria of interest that we'll talk about in a second. And then we had a NEC-like model, and we found that gestational stress worsened the intestinal injury in a NEC-like model in babies or offspring that came from mice that were stressed. So Lactobacillus is a anaerobic spore-forming bacteria. And the bottom bullet, it's a member of a family that produces short chain fatty acids. And so short chain fatty acids, that's a microbial metabolite that when certain food substances get to our colon, the bacteria breaks down those macronutrients into these metabolically active compounds. And so short chain fatty acids help to improve the intestinal barrier that Dr. Hunter talked about recently. And there's been a lot of papers about NEC and short chain fatty acids. And so where we are in our lab, some unpublished data is that when we're looking at babies who came from stress dams, we see a decrease in the butyric acid or butyrate, which is a major short chain fatty acid. And so this allows us to ask further questions in this space. So in summary conclusion, I think maternal gestational stress disrupts newborn immune function. And the mechanisms of how this happens in terms of short chain fatty acids are going to be needed to really understand and develop therapies for babies who are at risk for necrotizing enterocolitis. And it takes a multidisciplinary approach. And I think the NEC Society is spot on in this approach. In this space, we've had collaboration with psychologists, neonatologists, of course. We've had collaborations with families, also immunologists as well. And the family dynamic is so important. You know, once we know that once we can quantify the amount of stress during pregnancy, the big question is, what do you do next, right? And so that's a big question. And I think that answer is only going to be answered with the input from everyone, including families. Thank you very much. And I look forward to discussions at the end of my talk.
Thank you so much, Colin. Really appreciate that. All right, Joy.
Okay, can everybody see my screen okay and hear me okay?
Yep, perfect.
All right, well, I echo everyone else's comments. It's certainly a pleasure to be here, and I'm very happy to be a part of this panel. I was asked to provide a little bit of information about me and what I do, and then maybe a couple comments on our research, and then some interesting cases. And so hopefully I'll be able to accomplish that goal here. So just a little bit about me. I grew up in a small town in Pennsylvania called Hanover. I know we have a lot of people from in the United States and a few people from around the world in this call. I think I saw Bahrain on the chat here just a little bit ago. But Hanover is in the southern part of Pennsylvania on the East Coast. It's most known for its snack foods of all things. It's potato chips and Snyder's pretzels. And then there's a couple little diners throughout the city too that are pretty well known in that area. So after graduating high school, I went to college at the University of Delaware, which is in the northern part of Delaware in a city called Newark. It was a great experience, a beautiful campus. Majored in biology and medical sciences at that point. And then went to Penn State University in Hershey, Pennsylvania, sort of back to my hometown area for medical school. And for those of you who don't know about Hershey, it's where the Hershey chocolate bar was made. The actual, there's some actual pictures of the actual factory, which sadly was torn down a year or two after I left, and they built a brand new factory at the edge of town. But it's very interesting because you would wake up in the morning and you'd have the smell of chocolate coming from one side of town, and then the smell of burnt peanut butter from the Reese's factory coming from the other side of town, and sort of meeting in the middle for this kind of eclectic smell. It was kind of interesting. But after graduating there, I came out to Indiana, to Indianapolis, where I did my residency and fellowship. And then I stayed on the faculty here at Riley Hospital. It's been a blast. I've been here now for about 11 years, and it's been a wonderful, wonderful opportunity. This is just a snapshot of my family. Got several kids, a couple pictures here from our trip to Italy a few years ago. My wife, Sarah, and I there with Bentley, who just turned 1. And there's a little bit bigger picture of him there on the left side of the screen. So he is a handful, as I'm sure those with other little kids can probably attest to. So I think when it comes to, a surgeon scientist, a lot of people are like, well, what is a surgeon scientist? And, it really is, a physician or a surgeon who, really is involved in clinical or basic science research. Obviously, we take care of infants with necrotizing enterocolitis as a pediatric surgeon. And we do that in the operating room, which I think is a unique opportunity for us as opposed to other pediatricians, neonatologists, nurses, or dietitians. And then we also have a clinical or basic science endeavor. And as I said, I think the operating room experience is what sets us apart from others in the health field. Looking a little bit at my research, we all know that NEC is a bad problem, and we all know that there's not really a good cure out there yet for NEC. And so many of us here, have laboratories and research endeavors that are searching for a cure. We're searching for the next medication or the next device that will help babies with necrotizing enterocolitis. And so this is some early work in my lab. We focus primarily on stem cell therapy for NEC. So we're using stem cells either derived from the umbilical cord itself or from other tissues to treat necrotizing enterocolitis. And so the top panel here basically shows that when we give no stem cells to an animal with NEC, that we basically have a poor survival. So only about 30% of those animals survive. And then we have a dose response so that the more stem cells that we provide these animals, the higher the survivability. So we get up to about an 85 to 90% survival when we provide 2 million or so of those stem cells. And we wanted to check just to make sure it was the stem cell itself. So a stem cell is a cell that can divide indefinitely. It can differentiate into different types of cells. And the green bar here shows that it was the stem cell and not necessarily any cell. So a differentiated skin cell, for example, that was giving us our beneficial results. And we saw basically that the stem cells were really improving blood flow to the intestine, right? So, Dr. Martin showed a picture earlier of the bowel in our babies with necrotizing enterocolitis, and it was basically void of blood flow, right? It was what we call necrotic, so very poor blood flow to that intestine. And so when we added our stem cells, we saw that we actually increased the blood flow back to the bowel. So again, this is PBS is just like a saline solution that we used. The keratinocyte was the differentiated cell that we used, and then here's the stem cell. So when we start off, everybody's the same, like all the mice that we experiment on are the same. Red is very good or high blood flow to the intestine, and blue is sort of cold, no blood flow to the intestine. And so in a model of ischemia and reperfusion here, we basically clamp the blood vessel that supplies the bowel, and we can see that there's no blood flow there. And then when we release that, we see that we have some blood flow coming back. And then 24 hours later, we have a little bit more blood flow back to the bowel. And it was only the group that we gave the stem cells to here that we saw really an improvement, almost equivalent to what we saw at baseline. So that told us that the stem cells were really, responsible for improving that blood flow to the bowel. So we started to look at, what was it in these stem cells that was really promoting this vasodilation, and we stumbled on hydrogen sulfide gas. So hydrogen sulfide is sort of the smell of rotten eggs that you might be familiar with, and there's lots of beneficial, properties to hydrogen sulfide, and a couple of those include, decreased inflammation, maybe making more blood vessels to promote blood flow. But the most important thing seems to be its ability to vasodilate or relax blood vessels. And so we actually took, there's a way to silence genes in stem cells. And so we did that with the enzymes that make hydrogen sulfide. So we took the stem cell and we knocked out those enzymes that make hydrogen sulfide gas. And when we did that, we saw that NEC severity was much, much worse. So on the far panel here, we can see that this is our animal model with NEC, and then again that saline solution, that vehicle solution. So we have an abundant amount of NEC with a 10% high level of NEC. When we treated with stem cells, we drastically decreased the rate of NEC, just down to 20% and a very low kind of grade. However, when we gave those stem cells back with those knocked out genes, we saw that our NEC severity was much, much worse. Again, suggesting that hydrogen sulfide is a key chemical compound that's involved in stem cell therapy. So this is an interesting video. So we can actually take a blood vessel out of a mouse and kind of put it on a perfusion machine and look at it underneath the camera. And this is an interesting picture here. So we can actually contract the vessel. So we give it adrenaline or phenylephrine and contract it. And then when we add our stem cells in, we can actually see that this blood vessel dilates over time and kind of comes back to more of a normal caliber of a vessel. So this is sort of our hypothesis that we have in our lab. So we really think that there's injury that triggers intestinal cells to release a lot of pro-inflammatory mediators, so bad chemicals that kind of, signal danger, if you will, and that activates these stem cells to release hydrogen sulfide. And then we think that hydrogen sulfide works on the blood vessels to promote vasodilation, and then that works then to improve intestinal recovery. The other avenue that we're working on in our lab is a novel diagnostic tool. So again, not only do we not have anything to really treat NEC, we don't really have a thing to be able to diagnose it early or even predict it early. And so there's been a lot of push for some novel diagnostic tools. And so we've stumbled on what we would call an electronic nose. And these devices have been used in a lot of other areas over the years, including the agricultural industry, the petroleum industry, and really designed to kind of pick out unique compounds or unique smells within those samples to be able to detect impurities. And so much like, I kind of joke, much like if y'all seen CSI, right, there's a fingerprint, right? But within the fingerprint, there's all these little kind of components that make up the fingerprint, right? And so all these little dots make up a unique fingerprint. It's sort of the same way. There's lots of chemicals that make up a smell. And so this is a sort of a readout from our electronic nose, multiple different little smells that kind of make what we have dubbed the smell print. And so the thought is that we could use this device to be able to smell the baby's stool and to be able to detect if they have necrotizing enterocolitis. And so we've done that in both human and mouse models. So the top here is human stool samples, and we can see the blue is the NEC babies and the red is the control babies. And out of 32 sensors that we have, roughly half of them show a difference. So half of these sensors detect a difference between those samples. The bottom here is in our mouse model. And on the far left, you can actually see sort of a principal component analysis graph. So again, the blue dots are NEC mice. And then up here are the formula-fed control and the breastfed control animals. So you can see there's a distinct separation between those and the NEC animals. And that correlates too with a difference in the microbiome. So there's different bacteria in the mouse pups with NEC as compared to the controls. So it's great that we can detect it in a lab mouse or a lab rat, but really, what's the practicality of it? And I think that the idea would be, to use this data to build an algorithm to predict NEC. Would you be able to use this device to sense a difference in the smell of the stool 1 day earlier, 2 days earlier, a week earlier? It's hard to say what that level of cutoff would be, but our goal would be able to hopefully use this device to be able to predict which babies are going to go on to develop NEC. And we have people in our lab, we've dubbed them the stoomaliers, because literally they're going back and forth to our NICU collecting stool samples and analyzing them on this device. But our hope is that we can build this algorithm here to be able to move forward to predict NEC more readily. So the last part here of my little intro is just to kind of provide some special cases. I think, as a surgeon, there are certainly triumphs and then there are tribulations and pitfalls. And I'd like to end on a happy note, so I'll talk about the tribulations and pitfalls first. And I think for me, I've taken care of several babies, twin and triplet pregnancies. And it's really, really hard. It's hard for all the babies that we take care of who don't do well and who unfortunately pass away. But I think for twins and triplets, it's really tough, at least for me, and my colleagues on the call here can comment further. But it's almost for me like a Saving Private Ryan kind of thing, right? Where if one of a triplet passes or one of a twin passes, I really feel even more dedicated to that family and even more invested to try to make sure that we take the best care of those other babies as we can. And then from a success story for me, I had a little baby who was about 450 grams. It's the smallest baby I've ever operated on. It was so small I could hold him in the palm of my hand. And I had to operate on him about 3 times during his course here in our NICU. But he was a fighter, and miraculously he survived and he did very well. And it was an awesome opportunity, when he followed up with me back in the clinic at a year of age, our hospital did a great sort of reunion ceremony where they, filmed it and everything else. And it was really thrilling to see, somebody who was in such, so premature and in such a bad spot for the early part of his life to be able to survive and be doing well and to be thriving. So, every day I thank for the parents and families that are on this call. I thank you for entrusting us as surgeons to care for your babies. It's certainly a wonderful opportunity to be a part of the care of your babies. And, our goal again, I think, is just to kind of push the envelope forward and try to, develop cures and develop new diagnostic tools to help things further. So thanks very much.
Thanks so much, Troy.
Jen, can you see my slides?
Yep, it's perfect. Thank you so
Okay.
much.
Oh, wonderful. Thank you. Well, I'm the last of the speakers, it sounds like, and I'm thrilled to be a part of this webinar. So thanks for the opportunity to talk to you a little bit about my research. I'm Gail Besner. I'm the Chief of Pediatric Surgery at Nationwide Children's Hospital and a principal investigator in the Center for Perinatal Research at our research institute. And thinking back, the first baby that I ever operated on for necrotizing enterocolitis was baby boy Lewis. And this was when I was a pediatric surgery fellow about 30 years ago. He weighed 900 grams, which in those days was really quite small. And we operated for NEC and we saved his life. And I thought it was the best operation that I had ever done, but I was really naive back then. And I think I speak for all pediatric surgeons when I say that if we never had to do this operation again, and we put ourselves out of business for this one particular operation, no surgeon would complain about that. So I want to take the next few minutes to tell you a little bit about my research for necrotizing enterocolitis. And by way of disclosures, my collaborators and I are the scientific founders of a company called Scioto Biosciences. And I'll tell you a little bit more about that at the end of my talk. So I think we all agree that the etiology of NEC is multifactorial, but you've heard already a few people comment about the microbiome, and it's becoming increasingly recognized that altered intestinal microflora play a role in the development of necrotizing enterocolitis, which brings up the possibility of probiotic therapy for NEC. If you look at the percent of neonatal intensive care units in the United States that administer probiotic beneficial bacteria routinely to babies, it's only about 14%. But if you look at Germany, it's up to 68%, and New Zealand, 100%. So why is there hesitancy for probiotic administration? And I just want to thank Ravi Patel for permission to use this lovely slide that he made. So there are concerns regarding probiotic therapy use in premature babies. First, there have been multiple trials, but there have been variable results. The American Academy of Pediatrics, our largest group of physicians take care of children, encourages probiotic use, but only after Food and Drug Administration approval, and there are no preparations that are currently FDA approved. There's potential nosocomial infection from contaminants in the prep, and potential bacteremia from the live probiotic bacteria that are administered, and uncertain product quality. So whatever we do, we have to keep patient safety utmost in our minds. So we asked in our lab, can we improve the way we deliver probiotics to protect the intestines from NEC? In all clinical trials to date, probiotics have been administered in what we call the free living or planktonic state. But bacteria actually like to live in a biofilm state where they're surrounded by a self-produced matrix of DNAs, proteins, lipids, and oligosaccharides, in which state there's increased bacterial stability and increased bacterial activity. The probiotic that we're interested in is Lactobacillus reuteri, which is a gram-positive facultative anaerobic bacillus. It converts sugars to lactic acid, hence its name. It has antimicrobial properties because it can produce reuterin, and anti-inflammatory properties because it can produce histamine, and both infection and inflammation are important components in NEC, and it's capable of forming a biofilm. So our novel probiotic delivery system is really quite straightforward. We start with the free living bacteria, and then we incubate them with biocompatible microspheres. And when the bacteria, the probiotic bacteria, adhere to the surface of those microspheres, they start to make some biofilm, as shown by the green staining. But the system is very powerful, and we can load those microspheres with beneficial prebiotic substances, such as maltose or sucrose. And when we do that, it induces the bacteria to make even more biofilm, as shown here. So we asked whether enteral administration of Lactobacillus reuteri in its biofilm state could confer an advantage in protection of the intestines from NEC. We used a rat NEC model by which premature pups are delivered prior to term, and we subject them to repetitive episodes of hypoxia, hypothermia, and with the administration of hypertonic feeds and lipopolysaccharide, we see an appearance of the intestine that's reminiscent of the NEC that we see in the operating room. What we're looking at here is the incidence of NEC as determined by histologic grading. You can see the grading is from grade 0, which is no injury, all the way up to grade 4, which is severe injury. And in this graph, anything that's a shade of green represents no NEC, and anything that's a shade of pink or red represents necrotizing enterocolitis. So as you can imagine, in pups that are breastfed and uninjured, there's no necrotizing enterocolitis. In pups exposed to this experimental NEC, the incidence of NEC in this particular set of experiments was 61%. There was no benefit from giving Lactobacillus reuteri in its planktonic state, but significant improvement in the incidence of NEC when Lactobacillus reuteri was administered in its biofilm state. And it was noteworthy that these results were obtained with a single dose treatment prior to injury. Well, you've heard of the gut-brain axis, and this refers to a bidirectional link between the central and enteric nervous systems. The intestinal inflammation that occurs with NEC leads to the release of pro-inflammatory cytokines with activation of microglia, loss of oligodendrocyte precursor cells, a reduction in myelination, and this leads to the neurodevelopmental sequelae of NEC. So we next asked whether Lactobacillus reuteri in its biofilm state could improve brain function in survivors of NEC. Remember, we said that only about 65% of animals in this model die of NEC. Therefore, you can study the survivors and let them grow up. So we did several tests. I'm only going to show you one, the novel object recognition test, which looks at short-term working memory and the ability of an animal to explore a novel object and to memorize things. So here you can see the baseline results in animals that are breastfed and uninjured, significantly worsened results in animals exposed to NEC, no improvement in animals exposed to NEC treated with Lactobacillus reuteri in its free living state, but significant improvement back to baseline in animals that were exposed to NEC treated with Lactobacillus reuteri in its biofilm state. And we did several similar kinds of tests of memorization and found the same thing in all of them. And we asked what the mechanisms were by which Lactobacillus reuteri in its biofilm state is preserving brain function, and found that it preserves microglia, it decreases suppression of myelin basic protein, and it prevents the reduction of learning and memory related genes. So I don't have time to tell you all of our rat NEC data today, but suffice it to say that what we found is that compared to Lactobacillus reuteri in its planktonic state, a single dose of the probiotic administered in its biofilm state decreased the incidence of NEC, improves gut barrier function, positively affects the microbiome in NEC, and improves cognitive function after NEC. But as you can imagine, the premature rat pup is not really similar to a premature baby. But on the other hand, premature piglets are really very similar in size and other characteristics to premature babies. So we next asked whether Lactobacillus reuteri in its biofilm state could protect the intestines from NEC in a piglet model of the disease. As opposed to rats, where you have to subject them to a lot of stress, piglets, like human beings, develop necrotizing enterocolitis just upon being fed. So in our model, premature piglets are developed by C-section. Nasogastric tubes are inserted. They have to be subjected to some degree of colostrum to keep these premature piglets alive. In group 1, the piglets received this beneficial colostrum throughout the whole week-long experiment. And in groups 2 and 3, they were switched from a little colostrum initially to the administration of formula to induce NEC. In group 2, the animals were exposed to NEC but untreated. They just received saline only. And in group 3, they were exposed to NEC but treated with Lactobacillus reuteri in its biofilm state. And this is all enteral administration, I should say. And when we looked at the appearance of the animals, we found that the piglets in the NEC plus Lactobacillus reuteri in its biofilm state appeared clinically quite healthier than the animals in the other groups. And this led us to develop a clinical sickness score based on motor, verbal alertness, and other criteria. And what we found was that, as shown in red, the animals that were exposed to NEC but untreated had the worst clinical sickness scores, but the animals exposed to NEC treated with Lactobacillus reuteri in its biofilm state had improvement in their clinical sickness scores. We developed a definitive NEC diagnosis, and for an animal to have definitive NEC, 3 of the 4 following findings had to be present. And these combined things including gross injury score, histologic injury score, clinical sickness score, as well as bacterial translocation. And these are our piglet results. You can see that in piglets that received colostrum only, there was a low incidence of necrotizing enterocolitis. In piglets that were exposed to NEC treated with saline only, i.e. not treated at all, had a doubling in the incidence of NEC. And in piglets exposed to NEC treated with Lactobacillus reuteri in its biofilm state, there was improvement and decreased incidence of NEC back to baseline. And likewise, we saw a significantly decreased percent death due to NEC, as shown by the green bar compared to the red line. So my scientific collaborators and I are the scientific founders of a company called Scioto Biosciences in the hope of bringing this technology to the bedside. We've produced GMP grade, what we call SB121, which is the commercial name of this Lactobacillus reuteri biofilm formulation. This is suitable for human administration. And we did obtain approval from the FDA to begin phase 1 studies, but the phase 1 study approval was for adults before we might get approval for use in babies. The phase 1 study was actually completed in February of this year, and we treated 15 patients. We decided to treat patients with autism because autism is another disease that is very dependent on the gut-brain axis, and it turns out that over half of the patients with autism have significant gastrointestinal problems. We found that SB121 was safe and well tolerated with no adverse events, and surprisingly, we found that a significant percent of patients enrolled had a robust positive clinical response when you looked at certain tests that measure their degree of autism. This was unexpected, and we were very pleased to see that. So we hope that the safety data in adults, as well as our pig data, can be leveraged to go back to the FDA for approval for phase 1 trial in newborns. Again, of course, our long-term ultimate goal is to treat babies to decrease their chance of getting necrotizing enterocolitis in the first place. And I would just like to acknowledge all the research fellows in my lab that contributed to this work, as well as that in the laboratory of my collaborators. Again, I'm really honored to be part of this webinar, and I thank you very much for your attention.
Wonderful. Thank you so much, Gail, and to all of our presenters for your excellent presentation. So I think we're going to move right into our panel discussion. And Gail, I think you so kindly volunteered to moderate us.
Yeah, so thank you. As surgeons, you heard that we want to eliminate and eradicate NEC. However, we're confronted with the need to continue to operate on these babies. So we're going to go through a series of questions. And I'll just ask each of the panelists to, I think you can probably all unmute yourself because we're going to be talking a lot. You can put I think you all have your cameras back on, so that's great. And we'll just go, down the list. And if anybody in the audience has questions, please put them in the chat, and one of us is going to be monitoring the chat, and we can reiterate your question and see how the panelists would answer it. So the first question, and I'll tell you, these are very challenging questions, and there's not going to necessarily be a right or wrong answer to all of them. But the first question is, how do you know as a surgeon when to operate on a patient with necrotizing enterocolitis? So I'm just going to go down the list of participants. Kathy, do you want to give us a little insight as to how you do it in Oklahoma?
Sure. You know, I think that can sometimes be the tricky question, right? When to operate. I will say that the longer that I've done this, and also really, I think just spending a lot of time thinking about babies that have NEC, I am not in favor on waiting too long. Something that I hear sometimes is, oh, they're too fragile for an operation. They're too sick for an operation. And I tend to sometimes disagree with that, because at a certain point, if a baby really is that sick and they're not improving, it's kind of like a last chance. Now, I don't go in saying I'm going to fix everything, or I'm only going to go in one time and it'll be perfect. But at that point, I engage the family to kind of, so when I go through a kind of a discussion about surgery, I think there's a really good approach that's called, I don't know if anybody's heard of it, the best case, worst case scenario. It was actually an informed consent process that a vascular surgeon called Gretchen Swarze, who's a medical ethicist, came up with. And it's not really going through, you know, the usual risks, benefits, indications. It's really trying to paint a picture for that family of what happens in the best case and what happens in the worst case. So it kind of individualizes that experience or that outcome for that family, because numbers don't really mean anything. It's either 100% for your child or it's not. So I know I'm being kind of lengthy, but in general, there's clear indications, hard indications, like free air, you're going to operate. But for me, also failure to improve. I also get very nervous if I see a red abdomen. And I don't care to hear they're too small or too sick for an operation, because sometimes that's all you have left. And if I don't do something, then the inevitable will happen. And if the family want us to try everything, I will.
Colin, what do you have? Thank you, Kathy, very much. Colin, how about you?
Yeah, I agree with everything Kathy said. I would just sort of say when they've failed medical management, and so being on appropriate antibiotics, being sure their temperature is okay, bowel rest, appropriate ventilatory support. Once they have appropriate medical management and they're still not getting better or getting worse, that's when I consider an operation.
Mm-hmm. Yep. And Samir, do you have anything else to add?
I mean, I can add the simple, like, if there's air outside of the intestine, then that's an absolute indication. And then really it just comes down to, like, if somebody's getting sicker despite what you're doing, then you should do something different. And so that kind of pushes us to operate. So
Yep.
Just simplifying it.
Troy, anything else to add?
No, I think everybody covered it. I mean, you know, outside of the things that Samir mentioned, I agree with Colin. If they're getting sicker, if their ventilatory requirements are increasing, if they're getting more acidotic, you know, I think those kids deserve a look, because, you know, you could be missing something.
Yeah, Troy, are there any diagnostic tests that you can order that can help you with your decision making in terms of when to operate?
I mean, I think certainly the basic laboratory assessment, you know, looking for things like low platelet counts, alterations in bilirubin are helpful. Obviously, an X-ray, as you know, as Samir said, if there's free air, that's definitely a key finding. I think, you know, we tend to use ultrasound quite a bit here, particularly high resolution ultrasound, and I know that's sort of in the literature a little plus minus in terms of you know, its benefit, but you know, for those kids that we sort of are just maybe a little hem-hawing about whether we should operate or not, I think an ultrasound, you know, provides a little bit more information. And particularly, you know, only about 60% of babies who are perforated actually show free air on an X-ray. And so you're missing 40% of babies who are perforated. And so the key finding, at least for me, the key finding on an ultrasound to look for is fluid with debris in it. So if the ultrasound or if the radiologist says, there looks like there's complicated ascites or ascites with debris in it, to me, that's stool. And that kid's going to get an operation.
I love it. Anybody else use ultrasound in their institutions? And do any of the ultrasonographers use microbubbles? Do they inject anything intravenously to try to enhance the diagnostic abilities of the ultrasound?
At our institution, we use a lot of ultrasound at the bedside, and it's not necessarily just the radiologists that are involved in doing that. And I think we've gotten better at using bedside ultrasound for just diagnostic purposes in general. We use it for all of our line access, and we use it for identifying where the lines are located, say in like an ECMO patient. And so we use a lot of bedside ultrasound. And so I think that's been useful for us. The bigger thing for us is that with the ultrasound, if we see a collection of fluid, then it's allowing us to like sample that fluid and see if that fluid is somehow more complex, and that might give us a hint as to like go in and place a drain or get a more of a sample as to what's going on.
Samir, are you talking about paracentesis?
Correct.
So, Ann Kozlowski, who was briefly with us for several years at Nationwide Children's Hospital, decades ago, was the big promoter of paracentesis for necrotizing enterocolitis to figure out if babies had a perf. And I think we've generally gotten away from that. But are you saying that you actually do it?
I think we do it a lot for like, a lot of times what happens is you have a baby and they're sick and you don't know what's going on. And so you get an x-ray doesn't show pneumatosis, maybe it shows you paucity of gas, and that's where we find ultrasound to be more useful for us, where if we stick an ultrasound and we see like a large collection of fluid, then it may prompt us to sample that fluid, see if it's just ascites or if it's complicated. If it's complicated, then we then proceed with either a drain or a laparotomy. It's usually for us, ultrasound has been more useful in cases that we don't suspect it's NEC than it is in patients that we suspect it's NEC.
Any of the other panelists do paracentesis at your institution?
No. I think there's a couple of steps with that, right? Step one is if you're going to use the ultrasound, you better be pretty comfortable and good at doing it. And it is also operator dependent. So I think that that's kind of something that I would be pretty comfortable doing. I think that if I personally were concerned enough that there was some sort of complex fluid collection or fluid going on or a whiteout in the setting of kind of worsening or a non-improving clinical status, I'd probably be inclined to take a little look. But it's one of the things that I really like about being part of groups like this is that we have to keep questioning what we're doing and sometimes revisiting places that we think we've been, but maybe we haven't. So I'll be looking forward to seeing some of that data and publishing that out there because, Samir has certainly, given me some great advice in the past with some other clinical scenarios, and I'm open to it. It's just, it isn't something that I would offer right now. I thought there was a really good question that came up actually in our chat. There's a couple, I think Troy's looking at the QA, but in the chat, I think this was a really important one. From a surgeon's perspective, how long would you recommend that stage 1, early stage 2 NEC stays in the community NICU without access to ped surgery? And, I think for me, that's something that living now in a more rural state in Oklahoma, we just can't fit every potentially sick baby into a higher level NICU within our state. There just isn't that option. And I would, however, say you don't want to miss something, and sometimes the decline can be quite precipitous. So I wouldn't say stage 1. I think you're fine to keep where you are. But I think if you're entered stage 2, I think that's when you should probably start making some phone calls to see about getting them somewhere that does have surgical capability, just for that little bit higher level of attention, shall we say, and also the ability to quickly intervene if things do start to decline or fail to progress in a favorable manner.
Yeah, I think that's a really important comment, because when we learn as surgical trainees how to follow these patients, yes, you're going to get serial labs and serial x-rays and serial everything, but the most important thing to us is the serial exam. And we can't do a serial exam if the patient is not in our institution or somewhere where we can go and do it. So I think that's a really important point.
jump in here real quick from a question from the panel or on the Q&A was asking about serial ultrasound use and if that is something that's used to help with decisions to operate.
Do you guys mind if I share my screen for just a second?
No, that's fine.
So this is kind of what I use as like a algorithm. I kind of go like up here and I go, if somebody has a gasless abdomen, then those are the patients that I kind of use ultrasound for and see what's going on. If they have a free air, then they get an operation. If they're sick, then they get an operation. If they just have pneumatosis and nothing else, then typically we start with antibiotics and see where they're at. If that's helpful in terms of
Yes.
highlighting where we use ultrasound.
Before we get into the question of serial ultrasound, it's interesting you see that because I don't see that in the chat, but maybe I'm just not visualizing it. One thing we didn't talk about was serial x-rays, and I do think that this is something that's very powerful. And I have definitely had patients where you get, plain films every several hours, and there are fixed loops of bowel that just absolutely do not move. And this is highly suggestive of significant injury to those loops. So that is another thing that would, make me have a very low threshold of operating. And we shouldn't overlook the importance of getting a cross-table lateral film. Sometimes the nurses are a little hesitant perhaps to put the baby in a decubitus position, but there's no risk of getting a cross-table lateral film because we want to maximize our ability to see free air. So, Samir, what about the question about serial ultrasounds? Have you done that?
We don't typically do serial ultrasounds. We usually do them as a one-time almost diagnostic tool. I don't know what the data would show or what the right answers are, but that's just how we practice here. I do have a comment about the community level hospitals. So at Michigan, we cover about 5 different community hospitals as well as Mott Children's Hospital. So we travel a lot to the outside hospital settings. And we are not necessarily always called in patients who have stage 1 or early stage 2, even with access to ped surgery. Usually what we find is that if they do not have pneumatosis, they're usually treated with antibiotics. And if they're not sick, they're usually treated with antibiotics. If they have pneumatosis or are sick, by sick, I mean increasing ventilator settings, increasing abdominal distension, worsening acidosis, any of those findings that make you think, wow, this baby's getting worse, that's something that the earlier you can involve a surgeon or at least have a phone call with them, the better. I just don't think it's a good idea to wait a long time wondering.
Thank you. Let's move on to a really important question. How do you decide whether to place a peritoneal drain or do an exploratory laparotomy if you do have a baby that needs surgical intervention for necrotizing enterocolitis? Colin,
Troy, weren't you on the NEC study?
Yeah, I was. Yeah, so I think for me, for a long time, it was sort of just the size of the baby, right? So if babies were less than 1 kilogram, and they had free air, I would typically put in a drain. And if they were bigger than 1 kilogram, then I would be more comfortable to operate. I would say that, I had a mentor early on who is now retired, but basically, all the earlier studies would suggest that if a child was super sick and we didn't think would be stable enough for the operating room, that would be the child to put a drain in, right? And to basically try to see if we could get them over the hump, if you will. But that you would operate, in other words, do a laparotomy on maybe kids who weren't as sick. And this mentor of mine said in their mind it was just the opposite, and that's sort of like how I felt too, right? Like, so the kids who were super sick would be the ones that you'd actually want to do the laparotomy, because more than likely they were the ones that had a piece of dead bowel that needed to be removed or something. And the one maybe that just had free air, which is probably more commonly, spontaneous perforation, those would be the ones that you put a drain in. And so that's how I practiced, for the first 8 years or so, until the NEC study came out. And for those on the call who aren't familiar with that, it's basically a trial, a randomized trial that really compared putting a drain in versus a laparotomy, and compared outcomes in terms of mortality, but more importantly, which was sort of the big question was, how did that impact neurodevelopmental outcomes at a couple years of life? And the long and short of it is that if you just compare the two groups, drain versus laparotomy, the outcomes are essentially the same, right? So it doesn't matter whether you do a laparotomy or whether you do a drain. However, if the surgeon has and this is like statistical analysis that I am not smart enough for, but if the surgeon has a sort of gestalt that the baby has necrotizing enterocolitis, like so preoperatively, if they think the baby has NEC, then it's better if you do a laparotomy as opposed to a drain. So again, when you just compare the groups side by side, doesn't matter. But if the surgeon has the thought that it's necrotizing enterocolitis, then when they do the analysis that way, it proves that laparotomy should be done to improve outcomes.
That sums it up. But I have a question for you. Sometimes it's really obvious preoperatively whether a baby has NEC or whether they have spontaneous intestinal perforation because they have pneumatosis. So if you have pneumatosis, you have necrotizing enterocolitis. But clearly not all patients with necrotizing enterocolitis have pneumatosis. So let's say you don't have pneumatosis, you have free air. Do you feel in your mind that you're good at differentiating NEC from spontaneous intestinal perforation in the absence of pneumatosis? I'd like all of the panelists to answer that question. Kathy, can you tell the difference between NEC and intestinal perforation preoperatively in the absence of pneumatosis?
Not conclusively. I mean, you certainly have certain predictors of something being more likely to be like a SIP or a FIP diagnosis. Sometimes our more premature babies or smaller babies, they otherwise look pretty clinically well, and then they have actually quite an impressive amount of free air. Like the belly is full of a lot of excess air. It doesn't have that kind of whiteyed out appearance that Samir was talking about with reference to when to do the peritoneal, aspiration. And so those are going to be ones that I'm going to be thinking that's more of a SIP or a FIP, and probably more enthusiastic about a positive outcome with a drain on a child like that. Whereas if there's other strange features, the child's bigger, the child's older, they're less premature, they have some other features, including kind of a hazy opacity, they've been sick for a while, that's going to make me a lot more suspicious of NEC.
Colin, can you tell the difference between NEC and SIP preoperatively?
Probably not. Probably 50-50. The things that maybe point towards SIP, definitely agree with what Kathy said, but also if the patient has never fed, so if they've never fed before and they develop massive free air, that might tip me towards a SIP picture. But if they fed for a bit and they have free air, then it might be more towards NEC. But for me, my approach is usually about the same. My operative approach is about the same, but I don't feel like I'm great at definitely determining which is which.
Yeah, and that's what concerns me. Our center was also part of the NEC trial. And so what you heard from Troy was that if you just compare peritoneal drainage to surgery, the outcome is about the same for these babies. It's not great. But if your pre-op diagnosis is NEC, then the babies did a little bit better if you did an exploratory laparotomy and a bowel resection. And if your pre-op diagnosis was intestinal perforation, SIP, then the babies did a little better if you put a drain in. And the statistics that they used to determine this was something called Bayesian statistics, a complex statistical analysis. But I also, in the absence of pneumatosis, I also feel that I have a hard time definitively determining preoperatively which category the patient falls into, and then NEC trial results are dependent on the practitioner's ability to make that determination preoperatively. So if you put in a peritoneal drain, what motivates you to conversion to a laparotomy post drainage? Anyone can start.
These are really good questions. I mean, I think I was really know the answer, and it it's like if it's 7 p.m. on a Friday night and your partner is on call versus, you know, you're on call and it's like 3 in the afternoon. I think all of us do something different in practicality versus what we hope to do in theory. I think that if you place a drain, the thought is that that should be more or less definitive. Occasionally what we find is like you'll get an x-ray and there's still remnant air, or the drain isn't exactly where you want to be. It's actually only curled up in one small area. And so in those situations where you have a drain, but it's not like an ideal drain, you kind of wonder, gosh, maybe I just need to adjust this drain rather than to a laparotomy. And I think there's a reservation to like do something larger. If the person is not getting better because of inadequate drain, I think that's different than person not getting better because has disease process that's getting worse. And I think sometimes it's hard to tell, but in general, I don't think anybody would fault you to do a laparotomy and figure out what's going on in the belly, and actually look at the bowel.
Anybody else want to add anything to that? So I think what you're saying is, once again, if there's, you know, failure or progression to get better or continued deterioration after drain placement, that's going to push you towards doing a laparotomy.
I also feel that you have to look at our patients and our babies with the NEC, not in 24-hour settings. Like you look at these babies on 8-hour settings, that's a baby day, you know, 8 to 12 hours. And you have to be open to changing and modifying your plan and going a different way. I feel like sometimes I've seen cases where, well, the decision's been made to do a drain, so we're just going to do a drain and we're going to sit on it. And sometimes you have to change it or you have to escalate. And that's where sometimes I've felt like some babies haven't necessarily been given, I think, the best opportunity, because you have to be willing to convert, I think. So that's just one thought. There was a question in the Q&A that was actually almost like a little step back that made me think of a question for Troy as well. Somebody had asked for the benefit in predicting which babies will develop NEC with your research stuff that you presented, Troy. I was also wondering, does your fingerprint differentiate between SIP-FIP, NEC to kind of get to, you know, Gail's issue that we all have of not really being able to know sometimes? Have you looked at that?
Yeah, that's an awesome question. We haven't looked at that yet. We just don't have enough you know, kids enrolled yet to be able to kind of figure that out. But I mean, I think, you know, the question came across as, you know, what's the benefit of knowing or being able to predict which babies would develop NEC? And I think, you know, certainly if you had a sample or a stool sample that suggested that they might develop NEC, and then they go on to kind of have a picture like this where you're kind of confused, right? I think that could potentially be another data point to help you direct, you know, what type of therapy you would do. But I think more importantly, it's the use of sort of, you know, in the future novel therapies, right? Because you're not going to probably give, I mean, unless you have a, you know, well-defined probiotic like Gail's developing, you're probably not going to give, you know, like for us, stem cells or some other experimental therapy to every single baby in the NICU, right? And so being able to predict which ones are at highest risk, I think would be the ones that you would target for that. So, Jen, I wanted to kind of ask you, since we're talking about the drain thing, what's your thought as a parent? You know, when a surgeon comes to you, I mean, obviously, you know, drain versus laparotomy based on your involvement in this, but from a parent perspective, when you hear, hey, you know, we could make a big incision on your child's abdomen, or we could make a little tiny incision and put a little drainage tube in, how does that kind of come across to a parent or family member?
Yeah, I mean, I think certainly the smaller incision sounds less scary to the family, but I understand that might not always result necessarily in the best outcomes either. It just depends on the unique child. And I think, you know, from a family's perspective, I think a couple of things have come up is just how quickly the disease progresses and how fast we need to act to intervene, and those decisions of whether to operate or not, how consequential they are and how quickly they need to be made, and how overwhelming it is for a family, because I'm sure you all know this better than anyone, but the babies often go from being stable and then in a matter of hours, they're in desperate need of an operation. And so for the families to kind of have to quickly shift their mind to like, okay, my baby is doing well and is stable, to, oh my God, my baby's being rushed off to an emergency surgery operation, it's obviously overwhelming. And so I think, Troy, just your question of like which type of surgical kind of technique might be more appealing to families, while a smaller incision might be better. I think it's also important to just acknowledge that families are overwhelmed with all of the information that's being thrown at us, and like we are relying on our care team to help us understand that some things that might seem like more ideal might not be, and like helping families understand, the kind of long-term consequences of each kind of decision, and the benefits of the various options that are available to families.
And I think the other thing that's confusing that I've seen with some of my families is that, they're told one day, oh, your baby doesn't need an operation. And then the next day, oh, now we're doing an operation. And there's maybe not that much that's changed. I mean, because they're looking at their baby's right there. It maybe looks very similar. But sometimes time is the change. And if everything is still as it was from yesterday without an improvement, that in itself is another data point or something that would shift it. I've worked in, more than one kind of group, and then, sometimes, I'll come in with, a slightly, different approach, or that somebody will ask an opinion for somebody, like one of us who's maybe a bit more familiar with NEC, and our plan might be a little bit different. And I think that it's a huge stress, and it's a massive trust swing, I think, for families to kind of be on this horrific roller coaster of, one little thing looks better, it looks worse, and then suddenly we're pulling the trigger on surgery. So I have a lot of sympathy for that.
Thank you. I was at the American Academy of Pediatrics meeting a few days ago, and one of the presentations was on necrotizing enterocolitis, looking at large databases. And one of the points that came out was that there's a certain percent of surgeons that do a primary anastomosis when they're operating for acute neck. And I was wondering if any of the panelists do a primary anastomosis when they do a bowel resection, as opposed to making a stoma. And if so, can you tell us a little bit about when you do it, how you do it, etc.?
Yeah, maybe
go ahead.
Yeah, I'll start there. I typically do not do a primary anastomosis, largely just based on my training and my experience. The few times that I would consider it would be if the segment of intestine I'm taking out is really, really proximal. And I think the morbidity of a stoma so proximal or so close to the stomach would be pretty high. I usually don't do it just because all the things that go into an anastomosis not healing well, hypothermia, hypotension, acidemia, all those things, the baby may be evolving into that picture. So I typically don't do it, but be happy to sort of hear what other people do.
I was going to say that we actually do primary anastomosis now. We've kind of swung towards that a lot more. We've seen a lot of complications with ostomies, and like Dr. Martin was just mentioning, losing a lot of fluid can be an issue, especially if you have a proximal ostomy or if you've lost a lot of intestine. What we typically do is we don't do it up front. We'll usually do the bowel resection and then leave the abdomen open for several days. Go back in at around 48 to 72 hours, re-inspect the abdomen, see where things are. If we identify additional areas of the intestine that are either necrotic, ischemic, so forth, then we will address those. If the child is clinically improving and doing much better, then we may attempt a primary anastomosis. We almost never do one in patients that are below 1,000 grams. This tends to be in patients that are like, I don't know, 1,500 to 2,000, somewhere around there. Just so slightly larger babies than the ones that we typically will see with NEC.
Samir, when you do it, do you do a hand sewn or a stapled anastomosis?
We do a hand sewn. We haven't really done a lot of staple anastomoses in babies. I just don't have any experience with that, so I couldn't tell you right or wrong. It's just that in our group, at least, we do mostly hand sewn.
Yeah. The person presenting at the AAP, her data showed that there were 13% of surgeons in the United States that are performing primary anastomoses for NEC. And I followed up with the primary author. It was Jerry Golan from California, to make sure that those patients actually had NEC versus spontaneous intestinal perforation. And he confirmed that those patients had NEC, because well as you can tell from these kinds of databases. You know, I think sometimes it's hard to tell. Even in the NEST trial, I think that there was a little bit of, you know, difficulty differentiating what the patients were undergoing surgery for neck or for spontaneous perforation. Kathy, go ahead.
Yeah, I was going to say, I would suspect that maybe that database stuff is flawed, because I agree with what Samir said. I think actually you can do quite okay with a primary anastomosis as long as it's a second look situation. Those ones tend to do well. I can think of twice in 10 years that I've done a primary with an open and a close. The last one I did, it was exactly as Colin was saying. It was really kind of strange. It was this one focal area and it was incredibly proximal. The baby actually wasn't on any pressors. And I was really, you know, on the fence about this. And I did do a primary anastomosis and it did leak. And then we had a very proximal ostomy. And, you know, we did refeeding and they're all back together and fine now. But it, you know, I haven't had great success with it, except if you leave it open. And then I think they actually do fine. But even in the perfect baby, where no pressors or anything, I just think you're taking a little bit of a risk that just might not be worth doing. But again, that was an informed decision, and I did speak with the family about this as well to say, you know, this is what we're thinking and this is what we decided. So as long as everybody's on board and understands what you're looking for, that's the main thing.
Well, I know that Jen has some wrap-up slides that she'd like to go through with us. The time just seemed to fly by during this webinar, and I just want to really thank all of the panelists for giving us their surgical perspectives on neck. So Jen, we'll turn it over to you.
Thank you so much, and thanks for moderating. Colin or anyone else, did you guys have anything else you wanted to address in the Q&A or the chat box? Did we get everything?
I think there was an important question about the health disparity work that you were discussing. It was a person, I think, from the United Kingdom who published that they saw a study publishing that there was increased stillbirth and neonatal death, and it's still increasing in ethnic groups. And the question was, how can we get policymakers to start taking almost this institutionalized racism and this health disparity more seriously?
Yeah, thanks for that question, and thanks for restating it for me, Kathy. It's an incredibly great question with a really, really tough answer. I think as I think about necrotizing enterocolitis, and I alluded to it in my talk, is that so many things go into babies getting neck, or having prematurity, or having lung problems or heart problems, or neurodevelopmental problems. And a lot of it sort of focuses on, I think, the interaction with some of our vulnerable mothers and families to our hospital system. I think that just the discussion that you heard here, there's so many different opinions. And often when the family is at the bedside in these complex decisions, they participate in the decision making. But some of our families, for different reasons, they're not there, they're not able to be there, or they may not have the capacity to interact and participate in the decision making. And that sort of gets to a spiral of these already vulnerable populations, no input from the family, maybe not because of their own doing. And you have this widening gap of inequalities and poor outcomes between certain groups. So I think it comes down to a coordinated effort. So the things that I'm interested in, there's a lot of overlap with the pediatricians, the OBGYNs, the psychologists, sociologists. And so I think being willing to collaborate, being willing to be in societies like the NEC Society, where there's a large advocacy arm, to give a different perspective about what it looks like from the surgical side about caring for these really challenging patients.
Thank you. Thank you for taking the time to address that question. And we are grateful and honored to be able to partner with you all in advocating for equity and better outcomes for all of our babies. So I'm going to end with just a few wrap-up slides. I'd like to highlight how you can stay engaged with the NEC Society. We have a lot of resources on our website for both clinicians as well as patient families who have just been diagnosed, who have just lost their baby to this disease. So we hope that you'll check out the resources that are on our website. We have a blog that we update pretty regularly, so you can subscribe to that and stay in touch with us through our blog and on social media. We also have an in-person meeting coming up next year. I'll share the graphic for that in a moment. We are also going to be at a number of meetings coming up soon. The CHNC meeting, Hot Topics in December, PAS early next spring. So we would love to begin to see you in person now that we're back in person. And you can also support our work and help us build a world without necrotizing enterocolitis. So here is the symposium dates. It'll take place in San Diego. We're teaming up with Cincinnati Children's Hospital, but it will be in San Diego, not Cincinnati. And the calendar dates are August 28th through the 30th. And if you go to our website and kind of click a link, it's right there on our front page. I'll send you a calendar invite so the dates are blocked. So we hope you can join us in person then. The meeting will really focus on the research priorities that we've identified to be kind of the most meaningful and impactful questions and topics that we need to address in the field. So really looking forward to that. I also always like to share that we do have an online shop. You know, the holiday season is coming up. So if you're looking for something unique to give to people you love, or maybe in your lab or your institution, we have a lot of cool gear at the NEC Society's shop on our website to help us raise awareness about this disease. We've also recently launched a nurse ambassador engagement program so that nurses can become more involved in the NEC Society and really help us to advance our work. So you can learn more about that on our website as well. And just again, a friendly reminder that the month of October is Infant and Pregnancy Loss Remembrance Month, and tomorrow, October 15th, is the Wave of Light. So please join us in remembering infants like Micah, like Reed, like too many babies that have lost their lives to this really devastating disease. And I also have to briefly share that I am training for the California International Marathon in honor and memory of my son Micah and all of the babies and families that have been affected. And we're really excited. We have a team of 22 runners that will be running on the California International Marathon on December 4th. So if you're interested in cheering us on, you can do so. I'm happy to share that link with you on our website. And again, thank you to Sweet Reed's family for allowing us to dedicate today's webinar to him. And thank you to our really incredible speakers. We deeply, deeply appreciate just your commitment to families and to the field. So thank you all so much for being here, and we hope to see you again soon. Take care, everyone.
Thank you.
Thank you.
Thanks.